The topic here is mapping of genome using physical mapping. The techniques used: restriction mapping and FISH. Physical map is a representation of the genome of an organism as a collection of series of overlapping cloned DNA fragments or content arranged or ordered with respect to reference map, example a genetic map, as shown in the figure below. Physical mapping gives an estimation of specific distance in base pair between the genes. To construct a physical map, one should start with constructing a genomic library for the entire genome of the particular organism. The DNA fragment can be cloned into different types of vectors like YAC, BAC, depending upon the choice and length of the fragment that needs to be cloned. The different methods to construct a physical map is by restriction mapping, which includes both fingerprint mapping as well as optical mapping, fluorescent in situ hybridization or FISH technique, radiation hybrid mapping, and sequence tag site mapping or STS. Restriction mapping. The construction of physical map based on the relative position of restriction sites for a particular restriction enzyme on the cloned DNA fragment is known as restriction mapping. In this method, the cloned DNA fragment from the genomic library is digested completely or partially with restriction enzymes, singly or in combinations. The digested DNA mixtures are then separated on agarose gel to analyze and identify fragments of various lengths. The fragments are then arranged based on overlapping sequences to get the relative position of restriction sites. The main limitation of this method is that only small fragments of DNA can be used. Example: construction of physical map for 4.9 kb DNA clone using two restriction enzymes EcoR1 and BAMH1. Step 1. Selection of clone of 4.9 kb DNA. Step 2. Restriction digestion of clone DNA segment with the two restriction enzymes EcoR1 and BAMH1 singly as well as in combination. Next, analysis of DNA fragments on the agarose gel. The DNA molecules, when digested with EcoR1, gave two fragments of 1.5 kb and 3.4 kb. Note that the two values add up to 4.9 kb. Next, when digested with BAMH1, it gave four fragments of 0.7, 1, 1.2 and 2 kb. In combination, it gave five fragments of 0.2, 0.5, 1.2, 2 and 1 kb. Interpretations can be made after double restriction. First, if we take the zero point 0.2 and zero point 0.5 kb fragment. When put together, forms the zero point 0.7 kb fragment obtained or observed when digested only with BAM-H1, while when digested with both, it is cut into two fragments. Hence, it can be concluded that there is a restriction site for EcoR1 in zero point 0.7 kb length. Next, let us consider the 1 kb fragment in BAM-H1. It can be concluded that in this fragment there is no EcoR1 site as seen in the combination or double restriction result. The 1.5 kb length of EcoR1 digest fragment can be accounted if the arrangement is made as shown in the figure. Next, the 1.2 kb and 2 kb fragment has no restriction sites for BAM-H1 and must lie within the 3.4 kb fragment that is seen in the EcoR1 digest. Here, therefore, there are two possibilities as shown in the figure B. In such cases, partial digestion is done. Here, the 4 kb clone DNA is partially digested with BAM-H1. It means the reaction is stopped before completion by reducing the incubation time. This would result in only some restriction sites of BAM-H1B cleaved. Now, if the sequence in map 1 is correct, the two fragments 1.2 plus 0.7 should give a 1.9 kb fragment, while if map 2 is correct, 2 plus 0.7 kb should give 2.7 kb fragment for the partial digest with BAM-H1. The partial digest figure below shows the presence of 2.7 kb fragment. Hence, it can be concluded that map 2 is the correct sequence of restriction sites for the 4.9 kb fragment. In this way, maps are created for the clones in the library and then the overlapping sequences are arranged to give the physical map. This method of restriction mapping is known as fingerprinting method. The main disadvantage or limitation of this method is that most of the restriction enzymes have many restriction sites. This leads to formation of too many fragments, thereby making inferences becomes difficult. This can be overcome by choosing certain restriction enzymes which has limited number of restriction sites in the cloned genome, like the ones with longer restriction sequences, longer recognition sequences, example 7 to 8 nucleotide long, so that their occurrence is less in the genome. Example, restriction site for SAP1, SGF1. However, not many such enzymes are known. Another way is to use restriction enzymes whose restriction sequence contains certain rare motifs in the target DNA. Example, in humans, the 5-3-CG sequence is rare, as in humans, the 5' cytosine gets methylated. This makes it unstable and it undergoes deamination to form thymine. Thus, during human evolution, many of the 5-3-CG sequence has been replaced by TG. Therefore, choosing a restriction enzyme that can cut DNA at the 5-3-CG site would give unique long fragments. This would then increase the efficiency of analyzing the restriction fragment profile. Example, SMA1 enzyme, NOT1 enzyme. Another type of restriction mapping is macro restriction mapping or top-down mapping. In this, the chromosomes are cleaved using restriction enzymes with rare cuts. This would lead to the formation of large fragments of DNA. These fragments are then mapped and then ordered, as shown in Figure A. This type of mapping leads to production of few gaps in the map sequence. However, the resolution is low and specific genes may not be located. In bottom-down mapping or content mapping, detailed sequencing is done, but there are a lot of gaps. Combination of both approaches are used to create proper physical map or complete physical map. It should be known that the conventional agarose gel electrophoresis can only be used for separation of smaller DNA fragments. Fragments as large as 50 kb, the separation would not be proper because the migration rate is not linear and hence the resolution decreases as shown in the adjacent figure. Hence, the normal conventional linear electric current method cannot be used for separation of large segments of DNA obtained from rare cutters of restriction enzyme. This is overcome by using orthogonal field alteration gel electrophoresis. In this method, the electric field alternates between two pair of electrodes, as seen in figure A and B, each positioned at 45° to the length of the gel. The separation of the DNA fragments occur, but realignment occurs on alteration of the electrode signals. This leads to proper separation of the segments. Some other ways are using contour clamped homogeneous electric methods and field inversion gel electrophoresis. Another method of restriction mapping is by optical mapping. In this method, the restriction sites can be observed directly under a microscope. The steps include the attachment of DNA fragment to a glass slide so that they are stretched rather than being clumped. This is achieved either by gel stretching or molecular combing. First, the gel stretching method. In this method, the microscope slide is first coated with inactive restriction enzyme. That is, there is no magnesium ion present to activate the restriction enzyme. Next, the chromosomal DNA is suspended in a molten agarose and placed on the microscope slide. When the gel cools and solidifies, the DNA becomes extended. The slides are then washed in magnesium chloride to activate the restriction enzyme and cleave at restriction sites. A fluorescent dye is added for visualization. When these slides are examined under high-power fluorescent microscope, the restriction sites seem as gaps on the chromosome. This enables the relative positions of cuts to be recorded. Another method for stretching DNA for optical mapping is by means of molecular combing. In this method, the DNA fibers are prepared by dipping a silicon-coated cover slip into the DNA solution. It is left for 5 minutes to allow the DNA molecules to attach itself to the cover slip by their ends. The slip is then moved out at a constant speed of 0.3 millimeter per second. The force required to pull the DNA molecules through the meniscus causes them to line up, as shown in the adjacent figure. Air dried, the surface of the cover slip dries, retain the DNA molecules as an array of parallel fibers, and then it can be used for optical mapping. The figure here shows how a physical map is made or constructed by using different clones and arranging the overlapping sequences. Example, restriction sites. Another method of mapping genes on chromosomes to create physical map are by means of FISH technique or fluorescent in situ hybridization. It helps to create medium to high resolution physical maps. The basic principle of FISH is hybridization or binding of fluorescent labeled probes to complementary sequences in DNA, thereby helping in locating the sequence or gene on the particular chromosome. Generally, metaphase and interphase staged cells are used. Steps for FISH technique. The cells are first fixed with formaldehyde and mounted on slides and air dried. Two approaches can be utilized to denature the DNA. The first method, as shown in the adjacent picture. This is a conventional method where the probe and the cells are denatured separately and then hybridized. The mounted cells are incubated in the denaturation solution at 70 to 72 degrees centigrade for 2 to 5 minutes and then removed. Same way, the probes are incubated in the denatured solution. The composition of the two denaturing solutions may slightly differ. Then the denatured probes are mounted onto slides with the cells and allowed to hybridize with the cells. These cells are then incubated overnight at 37 to 42 degrees centigrade. After the hybridization, the cells are washed to remove unbound probes. The slides are then processed and viewed under fluorescent microscope and visualized. The second method is to denature both the cell DNA as well as the probe simultaneously. Here, first the probes are prepared. Probes are nothing but strands of short DNA sequence complementary to the sequence of interest in the genome. This labeled probes are mixed with the DNA fragment. The probe and the chromosomal DNA is then incubated at 72 degrees centigrade for 2 to 5 minutes to denature the DNA. This would allow the separation of the double strand of DNA into single strands. This is followed by the annealing step where the temperature is reduced to 42 degrees centigrade. This allows the probes that are used to go and bind to the complementary sequences present in the chromosomal DNA. The slides are then washed to remove the unbound probes. These are then viewed under fluorescent microscope to visualize the position of the bound probes on the chromosome DNA and locate the position of the sequences. Presently, fluorolabels with different colored emissions are designed. This enables to hybridize a large number of different probes on a single chromosome and distinguish the individual hybridization signals, enabling relative position of probes sequences on the chromosome. Example shown on the right for chromosome 17 for peppered moths. Thank you.